right, we are continuing our skeletal muscle topic with excitation contraction coupling. So for skeletal muscle fibers to actually contract, the membrane, that sarcolemma, must first be excited, which means that it has to have some sort of signal that is going to trigger a electrical reaction that is going to result in some sort of contraction. So this requires first being stimulated by an action potential, and this is always triggered from the nervous system for skeletal muscles specifically. So muscle fiber action potential is coupled to the actual contraction by the release of calcium ions from the sarcoplasmic reticulum, so that muscle fiber action potential is tied to the actual contraction of that muscle fiber through this calcium ion release. So all of our cells do have membrane potentials, which is a electrical difference across the membrane. So there's a difference in electrical current from the inside to the outside of the cell membrane. All cells have this, and there's various reasons for that, but in this particular case for skeletal muscle cells, this is absolutely essential to that contraction happening. So this signaling process comes from the action potential. And we'll talk a little bit more in detail about action potentials when we do the nervous system at the end of the semester, but an action potential is going to be a very, very specialized type of electrical signal that is going to travel through a nerve, in this particular case a motor neuron, so a nerve that is going to specifically result in movement. That signal, that electrical signal, is going to travel through the nerve and then end up going to these individual branches at the end of the nerve that terminates at the neuromuscular junction. And at that junction, the axon terminal, so the end of that nerve, is going to release a chemical messenger, which is also called a neurotransmitter. In this particular case, it is something called acetylcholine. ACH is the abbreviation for acetylcholine. These molecules are going to get released into that junction. That junction is an actual space in between the end of that nerve cell and the actual muscle cell membrane, so there's a little bit of a space there. And so this chemical, the acetylcholine molecules, get released into that little space, which is called the synaptic cleft. And they're going to diffuse or slowly move across that synaptic cleft and bind to special receptors for acetylcholine on the sarcolemma, so the membrane of those muscle cells. These receptors are kind of like locks that you would have on your house. So we have thousands of different receptors in our body, and each of them is specific to a different molecule, just like house and car locks are specific to different keys. So, you know, your house key is not going to unlock your friend's house and vice versa. Even though the same basic concept is there, you both have locks and keys, they're very, very, very specialized to each other. So skeletal muscle cells have these receptors for acetylcholine specifically. It's going to be located on something called the motor end plate of that sarcolemma, so the membrane of the muscle cell, which is on the other side of that synapse, that synaptic cleft. Once that molecule has bound to the receptor, there is going to be a special channel that opens, so a passageway that opens in the membrane, and positively charged ions, in this case calcium ions, are going to pass through into that muscle fiber, causing something called depolarization. And so that depolarization is basically a change in that electrical charge inside and outside of the cell. And so that electrical charge difference inside and outside of the cell is that membrane potential. And so for skeletal muscle cells, the inside of the membrane is usually 60 to 90 millivolts lower than what it is outside of the cell. So that's a pretty big difference for our cells. That's a really big electrical charge difference. And so when we move those calcium ions, those positively charged calcium ions, from the outside to the inside of the cell, it's going to bring that negative charge, that negative 60 to 90 millivolt charge, it's going to increase that closer to 0, and that's how we get that depolarization. As that depolarization happens, there are another set of ion channels or passageways that open called voltage-gated sodium channels. The name is pretty self-explanatory there. Voltage-gated means a change in electrical charge or voltage. We are working with millivolts in this particular case, and then this is specifically for sodium ions. And then these sodium ions are going to enter into that muscle fiber, causing that action potential, that initial electrical signal from the nerve, to rapidly spread or fire along the entire membrane to initiate this excitation contraction coupling process. So this is, this is kind of what that looks like. So here is our motor nerve cell right here. We'll talk more about the structure of these later in the semester. Here's our axon terminal here at the end, and the axon terminal is going to have all of these different little arms to it, and each of those arms is going to result in a neuromuscular junction, so a connection to that nerve cell. All right, at that sarcolemma. And so a more zoomed in here view in the middle. So there's that axon terminal there. You can see that space in here, that synaptic cleft, and we're releasing these little vesicles of acetylcholine that are going to go find those acetylcholine receptors. Zooming in a bit more down here at the bottom, and those acetylcholine is going to open that channel to have the different ions, calcium and sodium, moving in and out of the cell. So continuing talking about this excitation contraction coupling. So after we get that opening of those voltage-gated sodium channels, we need to keep this going throughout the entire muscle cell and muscle fiber. So for that action potential to actually get through to the membrane of the sarcoplasmic reticulum, there are those T-tubules that we looked at in the previous video that are going to allow a lot of those ions to move through that sarcoplasmic reticulum. And if you remember, there were a bunch of those little depressions in the membrane that connect to those T-tubules, and that's how we get all of that action potential to move throughout the entire muscle fiber. So that arrangement of those T-tubules with the membranes of the sarcoplasmic reticulum on either side of them is called that triad, and those triads surround that myofibril containing the actin and myosin. So those T-tubules are going to carry that action potential inside of the cell, triggering those calcium channels to open into the membrane of the sarcoplasmic reticulum, moving that calcium out of the sarcoplasmic reticulum and into the sarcoplasm, so that cytoplasm that's special for muscle cells. And that calcium ion influx is going to initiate the contraction of its contractile units or sarcomeres that we looked at in our previous video. We're going to continue this in our next video.